event level and transcript level, and I will explain later. And how do we study splicing? So this, for our analysis, we use the tool developed in our lab. It was called SUPA. And what it does, usually it just takes the TPM, transcript per million scores from different like experiments. We use TCGA, so FASTA sequences. You get the transcript scores for each isoform. And then from these isoforms, we separate at event level the psi value, which is percent splicing index. So how psi, this is the formula. Well, let's say that isoform, we want to know the inclusion of this event. We will collect the TPM value of the isoforms divided by all TPM values of all isoforms present for that gene. And this is how it looks like. So for example, here I'm showing you the tumor and normal psi values dispersed. And if you divide this, so this is breast cancer luminal A and luminal B. So if you divide tumor versus subtract tumor from normal, you get a score which tells you how much of this exon was included or excluded. So the delta psi value is if there is a inclusion or exclusion going on. So the lower the psi value, that means exclusion of the exon, higher the psi value, inclusion of the exon. So if say in this plot, here it is saying that the normal one have this exon highly included, while the tumors have this exon excluded or less included. Okay, why do we study at all splicing? So this is what is happening. If you don't study at a transcript level, what happens is, if this is one example of CD44 gene, if you only study at gene expression level, you see that the expression in all of these patients are more or less the same. There is no division. There is no difference between tumor and normal samples. But if you see the transcript level expression, you will see the inverted pattern happening. So there is in the tumor this isoform is highly expressed, while on the normal the other isoform is expressed. So that's why gene expression does not give all information. So we need to go back to the different transcript level or isoforms level to understand what is the shift happening here. What else? Alternative splicing is everywhere now. It's this. I want to just give you what more you can do with the studying splicing. So it can work as biomarkers. You can divide your samples, let's say, in different categories. There has been studies done also in our lab where just based on the splicing pattern, you can divide them in different clinical stages or different tumor subtypes. There are therapeutic uses of studying splicing. For example, here is one example of BRAF getting therapy resistance because of the lack of this RAS domain. So if the domain is present, the BRAF is the therapeutic target. But if the domain is alternatively spliced out, the BRAF doesn't respond to therapy, chemotherapy. It develops this resistance. Again, there are targets of therapy that can be used based on splicing signatures. So more you have splicing signatures particular to particular tumor type, more you can develop, let's say, therapeutic drugs that can be utilized. All of this is published in this review that we wrote, Eduardo and I, and it's interesting to read for where this is going, this feature of splicing. Okay, now my project. For me, I really like, okay, we are doing cool stuff, but can you explain your project to a 5-year-old? So I want to think how to explain my project to a child, let's say. So here, these are the surfers that are surfing in the sea, our different exon. And what we want is these surfers to arrive safely at the beach. Now, what are the components that are involved that can, let's say, harm these surfers? One, there could be something like a shark from the sea that can just come and eat them. So this here will be, I will call that cis component, that is like mutations, let's say, that are in the sea, like present in the sequence, that can harm or that can affect these exons. And second, like alien abduction, which is something from the outside is coming and taking or binding or taking them out. So here I will call them trans component, which is RNA binding proteins that are coming from the outside. They are not on the sequence, but they are affecting the splicing patterns. So basically, broadly speaking, my thesis was divided into two parts. First study is trans component, and second study is cis component to understand the regulation. So trans component, the first chapter was trans component, which was to study RNA binding proteins in different respects. What are the gene expression? What are their, where do they bind? Which tumors? Some RBPs are favored by certain tumor type, but not other tumor type, and things like that. This I'm not going to present today, but I have just put few slides just to give an overview. And second was, this is the second chapter, which I plan to present today, was the cis component, where we are studying all the sequences and whether the sequences have sites where these RNA binding protein binds, but they are not binding in certain tumor type because of mutations or some other, well, basically mutations. So to study this, we used multi-layer data from TCGA, which compose of all of these copy number variation, mutation, clinical data, RNA-seq data for different tumor types. And okay, first was to study the trans component, and then second was to study the cis component of the analysis. And just to give an overview for the first chapter, which is trans components, RNA binding proteins. This is the cancers that tumor type that we studied for these many samples. And what we've observed, the first thing was that the RNA binding proteins are highly dysregulated. So these are the different RNA binding proteins that we studied, and in different cancer types. And if you see, mostly they are categorized, we can categorize them by mostly downregulated, mostly upregulated. And then there were certain RNA binding proteins that had antagonistic effect, like opposing. So they were upregulated, let's say in lung cancer, but downregulated in certain other. So there were these patterns already we were seeing of RNA binding proteins having some kind of effect in developing tumor. These are the number of patients here where we studied. And sorry. Okay, so there are RNA binding proteins that are being dysregulated, but then do they have a binding site on these sequences as well? Because otherwise it doesn't make sense. So we studied gene by gene, also the binding site of this RNA binding protein. Let's say here I'm talking about MBNL1. So what I did is, this is the exon, and I extracted 300 base pairs upstream and downstream. Compared to control, I created this profile of if there is an enrichment of binding sites of these RNA binding proteins. So MBNL1, for example, if you see, the colored ones is for tumor, and this is upregulated in the tumor, in the breast cancer tumor samples, while compared to control, and is the same way for the downregulated for upstream and downstream. And this is what you see here. So delta psi is our splicing change index. So 0.1, like 10% of the patients are showing change in splicing in MBNL1. Sorry, RNA binding protein, this. And the downregulated upstream, they were MBNL1 was downregulated upstream and downstream, what we observed. This is another interesting, because what we discussed in the meeting also, I thought that it will be interesting to put, so I just inserted this slide, that based on this, we can predict actually correlation and anti-correlation between different tumor types. So here we are showing there are different tumor types here, and how these two are correlated. Like, let's say in this example, there is a kidney, this is kidney tumor, and if you see, this completely anti-correlated with prostate. So whatever behavior prostate cancer shows, there is an antagonistic behavior for kidney. Is the same for RNA binding proteins that were upregulated in prostate were downregulated in kidney. So it is so all of these correlation here, like higher the correlation means these two cancer types, like colon cancer and lung cancer, are very connected. So they have a similar protein regulation or RBP regulation going on. And certain, like let's say this one, kidney cancer and lung cancer, negative correlation. They are totally uncorrelated. So it was interesting study also to see like overview of the pattern of tumor behavior. And it is this one is another interesting plot for you, how you can. So it is not like one RNA binding protein, one gene. There is a network of RNA binding protein affecting the same gene. Some of them are binding upstream as a silencer and downstream as an enhancer, or vice versa. So this is, for example, different RNA binding proteins. These are the genes, and how they are forming you see like a network for each, all of them are working together. It's not like one specific is, we can specify later which one is the most important one, but otherwise there is like this network that happens. Okay. Yeah, so like just one example of NUMA1 or NUM gene that is known for proliferation gene in lung cancer, and NUM is regulated by quakin, MBNL1, MBNL2, and then ESPR2 and these proteins, RNA binding proteins. This study is published in Genome Research. Please feel free to Google and read the study if you want to know more. However, this is the topic that I wanted to talk today, which is the cis component of the analysis. This is published in Molecular Cancer Research also. Please feel free to look in detail. I will just superficially talk about it today. And also I wanted to thank Kusto for actually making me talk about this, because I finished this study and left science in a way. Like for 1 year I was not working in science. I went to work in a prison with like a rebel. I don't want to work here. But thankfully Eduardo published the paper and everything, you know, that was there. And then when I was making these slides, I realized that it was actually an interesting work. I should have talked about it more, because it's really, I mean, what we found with this study was challenging and interesting, and also not many people are doing it. So I think, so thank you for making me talk about this. So RNA binding cis component analysis, we are going to study the mutations on the sequences, but without any bias. So we are just taking our sequences and we are going to scan all the motif, all component, and we will see. This is why, because in tumor, it's not just, again, as I said, it works in a network. It's a complex machinery that is involved. This is one example of NUM protein that is known for proliferation in lung cancer. And is the same way, either the downregulation of this RNA binding quakin can have the same effect if same as mutations on RBM10, which is another RNA binding protein. So there are different mechanisms involved to achieve the same result. So this is why we wanted to, we could have finished our study but just based on my first chapter, which was transcomponent, but it was just a half story, because we didn't know about the mutation side, mutation angle. So that's why we got curious to make the second chapter. And for this study, we used almost 17 million mutations from TCG data from different tumor types derived from whole genome sequencing of 500 samples, patients. And yeah, so this is going to be the MIRA, the pipeline that I developed. What it does, it scans the whole, the genome sequence, the gene sequences, and then it tells you where these mutated regions are present, what is the sequence, region type, motif, presence on them, base changes, etc. So how I started with, I don't care with whether it is exon, intron, CDS, I don't know. I just have this sequence. I extracted from the gene for each gene, removing the pseudogenes. And what mutations, what is happening is, I don't think also that mutation falls precisely on exon, like this is the exon, let's mutate this region. It's mostly that this is happening, but the ones we study, we see more, I think, because people study WES sequencing, which is mostly just to sequence exons. So they don't sequence because whole genome data is expensive, and also our most of the genes, like cancer drivers, they have selective advantage because they are mutating the region which is making them tumor suppressors or oncogenes. So, but what we miss here is people like us who study on splicing, where the splicing regions could be on exon or introns nearby. So for people like us, we need to know, we need to extract all mutational information, not just the one that are present on, like, let's say exon, because exons are very short sequences. We have whole lot of things going on around. So this is what I did then. What I did then is like to take the whole gene sequence and then divide this by 7 mer windows. So 7, and then next 7, and then next 7, and then next 7, and then see what are the mutations falling on this region. So this is what I'm explaining here. We have k-mers of 6 mer length, and then we count the mutations on each of these k-mer. And we perform the binomial test and the correction for local biases. Binomial test is, let's assume that the mutations are falling everywhere, and then from there, what are the ones that are, like, let's say beyond certain threshold. And correction bias is, let's say that certain mutation is only biased for on A, just mutation is happening only on A. We don't want those mutations because that's a bias. We want all, so we removed all of these. We should remove all of these that have biases for the base pair. So with this statistical analysis, this is the pipeline that MIRA does. It takes the SMR, the ones that pass this test score, and then after we get our SMR, which will be significantly mutated region, it labels them where they are falling. So now then I go back and trace them, either do they belong in intron, exon, 5-UTR, where they belong. And then from there, once we have this significantly mutated region, now I want to know if there are binding sites present on these SMRs, if there are certain RNA binding proteins that are getting mutated specifically. So then we do motif enrichment analysis here, where compared to 100 control set will be just randomized mutation. Then we compare if there are enrichment of binding sites on these SMR. And then we can do downstream analysis, like whether these binding sites are affecting the RNA expression or the junctional change in the patients, in the samples. So this is the, the 6 main results that I'm going to explain. Let's start with the first one, which is significantly mutated regions. What do we see after our statistical test is, like, we get this score and we score with the p-value. And if you see, these are the regions that are already known, like BRAF, like highly mutated SMRs as regions. BRAF, all of the genes that you know as cancer drivers are here, TP53, PIK3CA. So this is saying that the ones that are known already, this is kind of validating our analysis. And then from there we select, so this is to select the cutoff score and NB score that I showed before. We do the analysis. This is from single mutation. So I run the k-mers for just one mutation, and then the k-mer with more than 3 mutations, let's say. And this is the score that we choose as cutoff, 7, because you see, we don't want just one mutation, because that's also a bias, that only one, if it is present in only one patient, one sample, we don't want that. We want at least it should be consistent, this mutation position. So then everything after 7 is the one that we are interested. That means they are present in either 3 or more patients. And yeah, then I map them back to different genomic regions, and we call them significantly mutated regions. And this is some of the examples of how they look like. So highly mutated BRAF, KRAS, for example, in CDS, in intron. You see, this is an NB score, the score, and the mutations of SMRs. This is 30 mutations on these regions, which is known. This is cancer drivers. The interesting thing is, there's the same 30 mutations here in intron, deep intronic regions that people don't usually study, because they are not as cancer drivers. They are not shown as cancer drivers, and the intronic mutations usually people don't study. So this was interesting that, you know, like, there are things happening here as equivalent as exonic regions. These are the number of SMRs we found. CDSs 209, and you see in introns there are like more than 1700 SMRs present. And then on 5' splice site, 3' splice site, which we expect to change in tumors. And also they were present on 5-UTRs and 3-UTRs. Second was, so now we have SMRs. Now we want to see whether these k-mers or SMRs are enriched on motif. Do they have a motif or RNA binding protein sites as well? So for that, this is, we took our SMRs, we created control regions, and then we did motif enrichment analysis using MOSI. This is a tool that I developed in our lab to do this kind of studies. And once we have enriched motifs, we labeled them with deep bind, which is a paper recently got published, which describes the RNA binding proteins and their binding site. So once we have the enriched motif, we use deep bind labels to label them. And this is the labels that we got. Here I'm showing for 3' splice site and 5' splice site. This is the labeled, the motif that we labeled. And this is the proportion of the patients where these SMRs or these motifs are enriched. Same for 3 UTR region. So same, these ones, also these ones are already reported in literature. So I mean, it's like a good validation that we are seeing this in our data. Same for introns. There are highly mutated RNA binding proteins like ZNF, PCBP1, HNRNP, L. These are, well, basically works as silencers in most of the tumor type, and we saw them highly mutated in intronic regions. Likewise, CDS and exon. This is all published in the paper. Please feel free to look if you like. And okay, now after that, because it is interesting that these SMRs have motif RNA binding protein sites and they are mutated. So third step was to see the conservation of mutations on motif. Like how does it look like? I think this is really, like, I like this results. Like one of my favorites was so this is the 6 MERS position of each base and where these are mutated. So this dark red is where they are mutated. So this G is mutated less than this G, let's say. And if these mutations are SNPs or real mutations, so SNP, we overlap this with the SNP databases to see whether, so the yellow ones are like no SNPs, the red one is that this position is known for SNP. We don't want SNP in cancer in the mutational studies because, yeah, because we are looking for somatic mutations. And then this is what then each base, how it is changing in what. So is there a favorable base change from this, let's say from C, it is always changing to G or always changing to T. So we wanted to study this also. So for each of these bases, we created another set of pipeline to produce this kind of plots that can tell you very precise picture of what's going on. This is another example for SRSF10 in CDS. So you see this G position, yeah, 4th position, is much like highly mutated than the other. So this is the favorable. This, and also if you see, this will break the motif. So if this is gone, the motif will break, which is interesting. That also this is converting from G to G. So SRSF10 or SRSF9 are known for having this tumor property. So it was an interesting result. Again, for 5' and 3' splice site, so you see GT and AG are the known core motif, which is here. However, there are mutations happening on the next sequence bases as well. Okay, so one thing is, because we are reporting many of these happening in introns, but are they meaningful? How do we know that they are meaningful? Because they are deep in the introns. What we thought that if these regions are meaningful, they probably will be conserved in other, let's say, other species as well. So what we did fast-fun analysis, the conservation analysis for SMRs, to see if these regions actually are relevant, like they are not changing in these different species. So which is, this is one way. What I want to explain here is we have SMR, but it's not that this SMR is present only if it is present in certain position. It will have high FASCON score. That means it is highly conserved in other species also. So we are reporting SMRs, but doesn't mean that this will be relevant in all positions. It will be only relevant in certain position where it will have property to affect different tumor type. So here is in 3' UTRs and non-coding exons. This is the Z score, conservation score, and some SMR distribution, and some SMRs are highly conserved only in specific site, not all sites that we are reporting. Again, the same, these are the FASTCON scores, so these are the different species where the sequences that we check, and these are the distribution of score that we get for different region types. Now, okay, after this, is it affecting? So this is these two things, this one and the sixth one, which is like new for us also, because people talk about mutation, this is happening, but they don't check if these mutations are affecting downstream expression way or not. So this was also important for us to, because we had the sample of RNA-seq data as well as whole genome sequencing data. So for the whole genome sequencing that we took patient, we also took the RNA-seq data of the same patients to see whether these SMRs or these mutations on RNA binding protein sites are having any downstream effect or not. So if they are meaningful or not. So here I'm going to talk about expression analysis based on the SMRs where these in the genes that were found. And for that, we took RNA-seq data. Salón is one of the tools which gives you TPM scores for gene expression data. And we, with the control matching control set, we created this analysis like a Z-score test for identifying the expression based on TPM levels of the patients. And we found that there were the SMRs, the TPMs, there were certain isoforms that were highly downregulated or upregulated based on where these SMRs were present. So some of these genes are interesting. This is in intron. And then likewise I have for exon and CDS, 5' SS, 3' SS as well. Somatic mutations in SMR show impact in RNA protein and expression. This is the same, but it's the same, it's saying the same idea, number of patients where this RNA binding protein site is highly mutated and how they are affecting on the expression. Third is the junction analysis. So not just the expression, it doesn't change the expression, but also we found that they also change the junctions. Like there are new junctions that are formed, new splicing variants that are formed based on these mutations around on these gene sequences. And for that we use Junkie, which is one software that tells you novel, if there are novel junctions present that I was talking. So here, yeah, you take this, well, this is the pipeline to follow, and then you get junctions new or known, and then you can cluster them, and you get the psi values, and so based on that you can tell if there is a splicing change happening or not in your data. So we did the same again with the control set, and we created the Z-score test, and this is again the same way of how the junctional changes are happening, like the expression changes. So this is the delta psi value, which tells you the splicing change index, and the Z-score, and you see there are genes that are getting spliced or creating new junctions based on the RNA binding protein and cancer type. So this is skin cancer. PDPP1 is the RNA binding protein site present, and this is the gene that is showing junctional variation. So likewise, this is for 5' UTR, but there are other in the paper about intronic or different regions. And this is these are again the same story. So these are RNA binding proteins, in which cancer type, and what are the genes that show junctional change in which. So this is, for example, breast cancer. All of these genes are showing different junction variants or splicing changes happening in breast cancer with RNA binding protein mutation in ZFP36. Okay, conclusions. So what we learned so far? That significantly mutated regions were found in non-coding and non-coding regions both. For us, crucial is that they are exactly the same way also in non-coding in intronic regions. So that means that it needs to be, these mutations needs to be seen deeper, not just on the interesting exonic regions. Second, these SMRs are enriched in putative binding sites. They have motif present, enriched motif present, and which can have their own downstream effect, like in change in expression or change in junctions. There are certain positions on the motifs that are favored for mutations than others. So maybe just one or two positions are where the nucleotide change is happening, which is going to break the motif, so that no RNA binding protein can come and bind there. Enriched motifs were found to be conserved in other species also. And mutations on the binding site show change in gene expression of the related genes with enriched motifs. So they do have a downstream effect. It's not that they are, so they are changing gene expressions. And likewise, they are also changing junctions, splicing junctions, because of these RNA mutations on RNA binding protein sites. And with this, I would like to thank. This is my group that I worked for, and thank you for your attention. Please. Thank you.
Thank you very much, Babita. And preguntas, questions, sobre todo los jóvenes. Sergio, do we have a microphone? No. Okay. Thank you for this interesting presentation. It's very interesting. I have a question. I don't know if maybe I missed it, but have you ever found any feedback between the dysregulation of isoform and the expression of the splicing factor that would regulate this isoform?
Yeah, so change in expression of the isoform, is it regulated with the change in expression of RNA binding protein? Yeah, this is the first paper, yes. The first chapter that I presented, it was all about that. So there are, yes.
So I have a simple question because I don't know if you, I couldn't understand if you explained that. So this mutated region that you found, for example, an specific mutation, you mentioned that they can alter the expression of the gene, right? And perhaps some genes that are related with the motifs that are associated.
Yeah.
But have you found also that the mutation of a specific splicing factor dysregulate the expression of other genes that are completely independent of these motifs that you said that are associated?
Yes, so this, I thought about it. So mutation on the splicing factor will not come on cis component because we are studying only sequences. But on the first paper, we checked for mutation on splicing factors and their effect on gene. Because first was like transcomponent analysis, and there this mutation on splicing factor will come. So we checked for if there are splicing factors that are mutated. There are mutated, but we didn't do, I think we didn't do this, what you're saying, the study between mutations of the splicing factor and the regulation of the.
This can be done?
Yes.
Okay.
Yes, based on correlation expression studies, because you cannot be very sure, like what the data that we have, you cannot be sure. Either you are, or you do the wet lab experiment and you take your gene of interest, and you know that this is influenced by this splicing factor, so you either do knockdown experiments and you see the expression of the gene.
Okay, thank you.
More questions? All right. So thank you, Rita, for your seminar.
Thank you.
Really impressive. I have a couple of questions from a general perspective, because you are working with a lot of data that are available out there from different studies. So I think for the general interest, how easy is to obtain those RNA-seq data, or are they really available out there? You can download them or use them. How fair is to use these data that are generated from other labs or from other studies and reanalyze and use them? Which is your experience or your point of view about that?
So for TCGA, if I understand, they are very easy to download. You just have to register, and I think this project was created so that the data can be shared in different community, in different but there was some embargo, if I remember, that you have to write them that can be accessed, and then they tell you, okay, till this time, or I don't know. But now for TCGA, what we use is all publicly available. If you are asking how clean the data is, like how is it too risky, that again, it's your personal, like people just sometimes they just take the data and start their analysis right away. In our lab, we made it sure that, okay, what we are going, first we have to normalize all the data, we have to remove if something looks fishy. And then we started our analysis based on the data set we found or we created, after the normalization. So it's, but people also say that these data that they put already in the public domain is already normalized or removed from artifacts and this. So you can also trust it and start doing the analysis as well.
And what about different databases apart from the TCGA? Are they some of them that are not from tumoral samples?
pathologies? I don't know,
There are,
obesity, diabetes.
Yes, yes. You will find, yes, because you have to search for databases or the publication, and you mostly, if they are you have published, they ask you to put your data set in public domain. So like Geo, I don't know if you know. So you there are several of these that are present now.
And they should be available and
Yeah,
accessible for
yeah.
all
I
of
think
us.
they ask you to make it public if you are publishing the paper.
Okay, thank you.
Any additional question? I may have a couple of them. One is very general and one very particular. The particular one is, have you implemented this kind of analysis that you have shown, or the first one too, in pancreatic cancer, adenocarcinoma?
Their pancreatic adenocarcinoma is included.
It's included in the paper. Because I didn't see it.
Then maybe it's not. Because I remember some of the cancer type were not available.
I was looking for it.
Well, yeah, maybe it's not then, sorry. Why? No. So there is glioblastoma, head and neck, kidney, lung, prostate, skin.
Well,
thyroid.
we'll do it.
Yeah,
we'll do it.
there is no.
We'll do
But
it.
pancreatic.
Not we, with I mean.
But it is in TCGA, pancreatic one. Is it in? It is
Yes,
in TCGA.
yes. And I think it would be very interesting because it would be very interesting if the let's say histotypes, the histology, the embryonic origin, kind of is squamous, adenocarcinomas, epithelial, etc. They have similarities in terms of splicing. If they have a common program that is shared also in terms of splicing.
Yes.
And the second question relates to something that Manuel just said, is do you believe, because this is not a cancer institution, some of us work on cancer, some other work on lupus or on obesity or reproduction. Do you think that this same kind of dysregulation, cis and trans, operates in other diseases and in other physiological circumstances, and that we just don't know about it and don't understand it yet, and that this kind of tools could be applied to understand them?
Yes, I think yes, because this is not, okay, we studied here mutations, right? And then we can say, okay, this is cis trans component. But at the same time, if you think SNPs are what is also this polymorphism that is happening, which is kind of probably changing the binding site, like mutations are changing the binding site, you know? So this is happening in other diseases, but we don't know because we are not studying, because they are large scale analysis and you need lot of data set to validate what you are seeing. So this one, because we had 500 samples for whole genome sequencing data, we could be sure what we are seeing is consistent, and that's why we are reporting that. Because otherwise, you see that I discarded all SMRs with just one mutation. That means it was present in only one patient. We don't want that. So it has to be at least 3 or more. At the same position, the mutation should be there. So that's why I think the limitation is this, and that's why there are not many studies done.
Okay, thank you. Any other question? Well, if not, thank you very much again for coming, and thank you.
Thank you very much.